Okay, I think we can start this symposium. Good morning to everyone. Okay, thank you. I have just prepared this slide to introduce the symposium. I would like to thank the British Journal of Pharmacology that sponsor our symposium and the Italian Society of Pharmacology for the financial support. And in this symposium, we will tackle the different biological pathways that underlie the pathogenesis of different neurodegenerative disorders. Indeed, it's quite clear that several primary neurodegenerative disorders are caused by protein misfolding and protein aggregation, but the mechanisms that contribute to the neurodegeneration are several and very, let's say, complicated. And to, let's see, put some idea in this context, in this framework, we also wrote a review that has been published in the British Journal of Pharmacology. And today, the same author will present their data. And so I would like just to introduce here our speaker. So we will have first Professor Jerome Mertens. He is an associate professor at the UCSD, so the University of California, San Diego. He did his PhD in Bonn. Then he moved to San Diego. And after the postdoc, he came back to Innsbruck in Europe, where he led his laboratory. But then this year, he just went back to the States. So he will show you his main results on the modeling of the neurodegenerative disorder in human cells, because his research interest is modeling aging and related neurodegenerative disorder using patient-specific reprogramming of the cells. And then we will have Daniela Mauceri. She is a professor in Marburg, and she has also a lab in the University of Heidelberg. And Daniela did a PhD in Milan in the lab of the Professor Monica De Luca. And then she moved to Heidelberg for her postdoc as an EMBO fellow. And in 2015, she started her lab in Heidelberg. And since 2022, she's professor at the University of Marburg in Germany. And she will talk about the structural and molecular mechanisms that control neuronal morphology and the relevance of these processes in the pathogenesis of neurodegenerative disorders. Then we will have our young investigator. It's a pleasure to have here Roberta Facchinetti, that has been selected by the organizing committee. She is a young postdoc in the lab of Professor Scuderi at University La Sapienza in Rome, and she got her PhD in 2019 in pharmacology and toxicology. And she will present this oral communication on oligodendrocyte astrocyte crosstalk required for myelination in Alzheimer's disease. Not finally, but we will have Alerie Guzman, and she has a lab in Alicante, in this Instituto de Investigación Sanitaria y Biomedical in Spain, and Alerie did her PhD at the University of Cambridge, and then where she did also her first postdoc, but then for the second postdoc, she moved in Belfast. And now in 2022, she got this Miguel Servet Investigation Award to set up her group in Alicante. And she will focus on the role of the oligodendrocytes at the crossroads of inflammation and neurodegeneration. This is her main interest. And then we will have my presentation. I'm Elena Marcello. I'm an associate professor at University of Milan. I did my PhD at University of Milan in Professor Monica De Luca lab. And then for my postdoc, I went to Pamplona in Spain in Isabel Perez-Otaño lab. And since the beginning of my career, I focused my attention on the mechanism that controls synaptic failure in Alzheimer's disease pathogenesis. But now I don't want to steal more time to our presentation, so I invite here Jerome for the first lecture. Thank you very much.
Well, first of all, thanks a lot, Elena, for organizing the symposium to bring us all together, and I'm happy to see so many faces still here on the last day. This is just a snapshot of the review we wrote. So I'm going to spend the next 30 minutes talking about how novel approaches for modeling neurodegenerative diseases in the context of aging, and how we use these to identify new potential targets for treatment of age-related neurodegenerative disorders. Aging and neurodegeneration are closely linked, and I think Alzheimer's disease is like a prime example, which is known to be highly age-dependent, in that even very aggressive early onset familial Alzheimer's disease patients only develop the disease when they reach the age of end of 40, 50, which is of course way too young to become demented, but it still shows that there's 40 or 50 years of proper brain development and function before that. So age is definitely an important risk factor here. And what many, especially basic scientists, don't appreciate enough, I think, is that the vast majority of Alzheimer's cases are actually sporadic, which means they develop at the absence of very known mutations in the APP processing pathway, for example, in APP or presennin or gamma secretase proteins, making age, for the vast majority, the most significant risk factor for the development of AD. And what's also known is that AD is somewhat sneaky, in that symptoms really develop over years and maybe decades before the manifestation of dementia, indicating that there are a lot of cellular and molecular mechanisms going on in our brains before actually somebody gets demented. And what we think is that in any proper treatment that could halt, reverse, stop, prevent the progression of a mild cognitive impairment, for example, into Alzheimer's disease would have to start really, really early, because it might not be really reversible to regrow the brain cells and have them function in the same way. And with no unifying disease etiology, one pathway known that causes this disease, we don't really have any treatments or cures at the moment. And the way we and other labs try to wrap our head around this is that we're thinking of a multiple hit theory here, where one hit certainly comes from your genotype, so there are complex genetics involved, it's humans, but also the environment and the lifestyle someone is living. But one essential hit comes from aging, and that we think that these hits meet at certain convergence pathways in our brain. These might be actual signaling pathways, cell-cell interactions, aging cellular structures, for example, and then these come together, reinforce each other, and lead to the dysfunction, disintegration of neurons, degeneration of neurons, and then ultimately the age-related disease such as Alzheimer's disease. So the questions we're asking and trying to answer in our lab is, number one, what are the differences between a young neuron, an old human neuron, and a neuron from a diseased patient, and then try to figure out in these model systems how and when do the multiple risk factors for Alzheimer's disease, for example, and aging really converge, and what can we do about this? So for me, being a trained cell biologist, a stem cell biologist, the ideal model system obviously would be to have living neurons directly from the brains of young people, healthy old people, and patients, and just take them in the dish, throw all our cell biology and multi-omics technologies which we have today on them, and figure out what's the differences. And obviously we can't really do that, and we don't really want to do this. So we have to create these cells or cellular models in another way. And the go-to system for this would be in the field iPS cells. So you know that induced pluripotent stem cells can be generated from different types of somatic cells, for example, blood PBMCs or fibroblasts, basically from virtually any stage of human development and aging, and can be reprogrammed into these iPS cell cultures, which largely resemble the inner cell mass of the human blastocyst, which is approximately 8-9 months before birth. And these cells are pluripotent in that they can generate virtually any cell type of the human body, liver, pancreas, but also brain neurons. And these are obviously a very interesting model system, as they are human neurons and they capture the whole genetic makeup of a certain patient. However, the issue with a very age-dependent and kind of non-genetic or genetically not well understood disease such as Alzheimer's disease is that this iPS cell reprogramming process, already in the very early steps of this process, completely rejuvenates cells. So it erases the aging information on the protein level, on the epigenetic level, transcriptome level, which is great news for people that want to use iPS cells, for example, for cell replacement strategies. There are clinical trials going going on for Parkinson's, for example, in Japan and the US. And this is great because these spare part cells are very young and fit cells, just like basically like in the embryo. But for us, a lab that wants to model an aging or age-dependent condition, this is obviously a big limitation, which is why we're mostly focusing not on iPS cells, but on the direct conversion of somatic cells, specifically fibroblasts from the skin, into so-called induced neurons, or our INs. And the 10,000-foot view on this is kind of similar to iPS cells, so we're also overexpressing transcription factors, just not pluripotency-associated transcription factors, but pro-neuronal transcription factors, in this case, for example, NGN2 and ASCL1, which turns fibroblasts around with a pretty good efficiency of like 60, 80 or more % into functional neurons within 3 weeks, and then you can leave these cells also to mature in culture. And just over the past like 5-8 years, we and several other labs have shown and confirmed that broadly, in contrast to iPS cell models, that INs are largely age equivalent. And with age equivalent, I basically mean two things here. Number one, they should have the biological age of a donor, so like a neuron from an 80-year-old should be different to a 20-year-old in some ways, and they should possess an adult cellular identity and not a fetal identity. And just like how we think of these two things in data is looking at the ontogenic identity of a cell. One thing you can do is to look at the whole transcriptome signature and compare it to like a database that's out there. So we're lucky that we have the Allen Brainspan Atlas that has like hundreds of samples from the developing human brain there, from 8 post-conceptional weeks up to birth, and then also several old and now also some aging samples. And what we did here, we took several transcriptome profiles from INs and also from iPS cells that we generated from the same old patients, which you see in the darker blue here, or darker green, and then just look for similarity. And what you see is that the iPS cell neurons really resemble the fetal stages here and are very similar to those, but then don't really show a high similarity to adult brain. And this is really where the IN model shines, and that it's not really reflective of the developing human brain or brain neurons, but then is more similar to the adult human brain, which we think is a very good reason to use these cells and to look into patient-derived cells, what's actually going on. And then the other feature about age equivalence that I mentioned is the biological age, and there's again no gold standard to really measure this. One very broad and integrative, useful way and quantitative way to look at this is to look at epigenetic age or epigenetic clock, DNA methylation clock, which you have probably heard about. So just in a nutshell, you can look at a whole genome-wide DNA methylation landscape, and there are algorithms that, based on just like 300 to 400 CpG islands and how their methylation status, can predict a person's or biological sample's donor's age. And we did this for our fibroblasts that you see up there in blue, from a cohort of like 60 to 90-year-old people, and in these fibroblasts we see the biological epigenetic age, and then when we reprogram these cells or program these cells into INs, they actually show the same epigenetic age, indicating that they remember from which patient age they came. However, when we reprogram back into iPS cells, you see this down there in purple, and even if we keep these iPS cells in culture for a long time, like a 6-month culture experiment is pretty long, but we're still seeing that these cells show pre-birth ages, indicating that they actually do what they should do, right? They are the inner cell master of the blastocyst, and they should develop into a human that gives birth after roughly 9 months. So this was way more enough information for us to actually take this model system one step forward and to create these INs from a cohort of non-demented but aged control donors, as well as mostly sporadic, also some familial AD patients. And just in the immunostainings down there, you just see that these cells properly form neurons from all patients and controls. We didn't really see any differences in how well the fibroblasts convert into neurons. And then when you just look in the dish, these cells after conversion, say after 3 to 5 to 6 weeks, it's virtually indistinguishable from like primary neurons or iPS cell-derived culture. So these cells are neurons and they are functional. So to make sure that really the fibroblast from the AD patients and the controls converted in the same efficient way, we again took their transcriptome and looked at similarity to the IN conversion trajectory, like at different time points from fibroblasts on their way through intermediate steps into what we could consider fully converted neurons at 20 days. And what you see here in the red orange colors is that the similarity of fibroblast obviously drops sharply after this conversion is initiated, and it's similar for AD and control fibroblast, and then also similar when we look at the IN transcriptome profiles, they obviously become more similar to the neurons that we're generating and lose similarity to the fibroblast, and we don't really see any difference between ADs and controls. So what's going on there? And obviously we could have looked just at like aggregates, we could have looked at tau, we could have looked at amyloid beta, but really this is mostly inspired by very late stage pathology and by the genetics. And we wanted to get an unbiased view on this. So we performed whole transcriptome RNA-seq on all these patients and controls, looked for differentially expressed genes, and there are many ways to look at this. Here in this heat map you can just see the most significantly up and down regulated genes, and one thing you can appreciate that this looks pretty good, but there are also outliers here, and this is just where you have to appreciate that each cell line actually corresponds to one donor, so it's one person, and just look around you in the room. I mean, there's quite some variability between us, even though we have been classified if we're AD into the AD category or not. And this is why, like a cohort of where we are pretty proud of, like over 30 samples, which is at the time was like one of the biggest studies. Is really just the beginning, and we have to look at more people if we want to do these endogenous human studies. So to just make sure that we're looking not as just like artifactual things, we're not the first people that look at a transcriptome from an AD human sample versus a control. We took this data to overlap it with a lot of data sets out there, from microarrays to RNA-seq, from hippocampus, cortex, and so on and so on. This just gives you a very brief overview here in the pie charts, is that usually we see a very big overlap and a more significant and increased overlap with the AD signatures that have been reported before in different studies. And I think what's most useful is one thing to also compare to single-cell RNA-seq data that just came out from patients and controls. They don't have as many patients and controls as other studies, but they can see the different cell types and then the AD-specific transcriptome signatures in the different cell types. And when you look at this little heat map here on the right, you see that our IN model, which obviously only has neurons, we have no astrocytes, we have no microglia, we have no vasculature, and so on, shows a high similarity to only the neuronal AD signature, but not to the oligodendroglial or astrocyte signature, giving us quite some confidence in these data to move forward also to analyze this in an unbiased way. So we did this and looked at the downregulated genes first, indicative of what cellular functions are downregulated in these cells. And there's not so much surprise here in that we see many mature neuronal gene sets. So you see that in the semantic space, but picked out some key pathways here for you. If they move to the left, they're downregulated in the ADs, and many of these genes relate to mature neuronal gene sets, neuronal activity, channel synaptic axonal function and maturation, which is not that much of a surprise since we're dealing with a degenerative disease here that's characterized by the loss of neuronal integration, loss of neuronal functionality, and eventually degeneration. Since we have living neurons in the dish, we can kind of go forward and confirm these with functional or cellular assays here. And just to like put a check mark like on each of these categories, we could see that the ADINs actually have lower synaptic densities than the controls in culture. They form less complex branching morphologies, and they also show by live cell calcium imaging decreased spontaneous network activity, which again kind of inspires confidence in the model. So we felt good when we moved forward into looking at the upregulated gene sets, so kind of like what pathways are activated that are causing this, or what is the cell trying to do to combat these. And that actually took us quite some while, because like you see that semantic space is quite crowded. You see a lot of Go terms and KEK pathways to be upregulated, and they don't really like show many of the things you might expect. So starting easy, some things we see are expected, and that we see cellular stress and damage coping pathways to be upregulated. But then the vast majority of what we see really aligns to the upregulation of cellular signaling pathways that we know from development. We know them from stem cells. There are stem cell-related signaling pathways, for example, neural stem cells, but also pathways that are very, very famous specifically in cancer. And this is something that didn't really make a lot of sense to us in the beginning, because we're dealing here with properly post-mitotic neurons in the dish, and they don't perform any like cell division, and there should be no discussion about cell division within the expressed genes of these cells. So it took us a while, but overall the signature was quite clear with like hippo signaling. You see notch signaling, HIF1 alpha, cell proliferation, both pro and negative regulation, p53 oncogene pathways to be upregulated in these neurons. And this all indicated that these cells somehow have lost their mature neuronal identity. There's something going on that's non-neuronal. It's stem cell-like and it's cancer-like. To just confirm that some of these like addressable changes that we see are actually happening in the cells also on function and protein level, we looked again. We could confirm that the cells show actually increased reactive oxygen species levels and DNA damage or damage repair signals in the cells. And they also really start to express non-neuronal proteins. So for example, LDHA is a glycolytic enzyme that produces lactate in the end, and this is a metabolic pathway that neurons really don't want to do. They do want to do oxidative phosphorylation, and I would get into detail a little bit later on this. And they also start to express cell cycle markers such as cyclins, for example, which usually shouldn't be expressed in neurons at all. Just as a disclaimer, we spend a lot of time actually trying to find a neuronal cell cycle in our cultures in the ADINs, but despite them expressing cyclins, for example, we never really saw this. So there's not like a real cancer kind of proliferation of neurons going on, but we do see these immature signals to come up. So something is going on in these cells. So overall, our hypothesis here was that these cells, the ADINs, seem to have kind of lost their mature identity. They are locked in some kind of a hypomature, unnaturally immature, dedifferentiated state. To look at this again in the transcriptome, we did this kind of mapping approach again, but this time not from the fibroblasts to the neurons, but actually from human iPS cell-derived neural stem cells, NSCs, and then over different time points into neurons. So neural stem cells and these immature neurons are stages that during the IN process from fibroblasts to neurons, you never go through a stem cell. You go from a slowly proliferating fibroblast into a post-mitotic neuron, and that's it. There are no signals of stem cells coming up. However, when we looked into AD and control INs from this cohort, is that the control INs actually behave the way you want it, right? So they show the highest similarity only to the most mature stages of neuronal differentiation, while the ADINs, just in their whole transcriptome identity, seem to be somehow confused on that scale and also show similarity to immature, even neural stem cell transcriptome profiles, indicating that these are really not fully mature neurons, but not because they didn't convert, but because they, the stress and the response of the cells to the stress that we see in culture probably led to a dedifferentiation of these cells. To see if this is just like an acute change in the transcriptome, like, hey, the cell decides I need like these 5 proteins here, let's make some mRNA, and we're looking at mRNA abundance mostly. We had to look also into the epigenetic landscape of these cells, because this is really what's defining the identity of a cell. And this is quite useful to know that progenitor stem cells, but also cancer cells, globally have a very wide and open chromatin landscape, making them flexible to, for example, differentiate into different cells, while a fully differentiated specialized somatic cell would have a very densely packed, very defined chromatin landscape. And this is one thing you can look at with ATAC sequencing, where you just profile and sequence for open regions. And I don't want to go into detail here to spare some time and also go more like to the target kind of identification in this, but just leaving you with this volcano plot where each dot represents a significantly a significantly differential open region in the chromatin. And if it's on the right side, it would be a peak that's more open in the AD-INs, and if it's on the left side, it's more open in the controls. And what you see basically is that the enriched openness in the AD-INs is just a global thing. And then you can take each of these stretches that are more open and look to like what kind of promoters are they associated with, which kind of transcription factors probably bind to these sequences. And these data kind of make a full circle back to these signaling pathways that I showed before, MYC, Notch, HIF1 alpha, P53, and so on, are really like epigenetically more open, making these cells in their identity less mature but more stem cell or cancer-like. One question, like, did we really have to do this? Is this really something where we need to use INs, or could we have just like used iPS cells? So is this phenotype actually dependent on age? And if so, how much of the phenotype depends on age and how much is maybe still there if you rejuvenate the cells? And this is really like a beautiful thing we can do with the system, just when you want to spend a lot of time and money, reprogram all these fibroblasts again into iPS cells, differentiate them into neurons in a very similar way to the INs, and then compare what's left, what's the role of aging in this model system. So this sounds quite complex, but the answer we got was pretty black and white, and that we see absolutely zero significant differential expression between AD and control iPS cells, which is not surprising, but also not between the iPS cell-derived neurons. Here you have to, little disclaimer, it's a little reduced cohort of just 10 against 10, but also when we're reducing the IN data to 10 against 10, it should give us a lot of significantly differentially expressed genes, indicating that this phenotype is really dependent on age. With this, I don't want to say that you cannot model familial AD with iPS cells, and not even that you cannot model sporadic AD with iPS cells. It just takes a little bit more than just making these cells for 3 weeks and looking what's there endogenously. There are strategies to age these cells, there are strategies to stress these cells, and many really elegant stories have used this paradigm. And actually, when you look into our, like now, Jerome's favorite pathways here that drive this cancer-like dedifferentiation of the cells, here now in light blue, we see that, again, no gene in these volcano plots, in these violin plots, is really significant, but they all pathways seem to tend to be more upregulated in the ADs versus the controls, indicating that also in iPS cells, if you just age them or stress them in a proper way, might actually show the same, what we would consider a flag post-central phenotype of Alzheimer's disease in the dish. So with this, I would get to the summary of this part one. So with this last piece of data showing that this phenotype really depends on age in your system, in your human model system, but it seems to be distinct from normal aging, and that we see not the same genes that we see just in healthy aging, for example. I haven't really shown this data. And most importantly, that the AD phenotype in these cells actually doesn't seem to be just like toxic proteins or aggregates build up and just kill a cell, but these cells, in response to damage and stress on a chronic level, start to drive and activate and drive a malignant cellular program of cellular dedifferentiation, leading to a loss of identity and locking them in this hypomature state. And this hypomature state, with this loss of mature protein expression or gene expression related to synapse, to axons, and so on, is well suited to explain the loss of neuronal integration and functionality, which predates the actual death of these cells during the disease. Honestly, we're also not the first people to describe, for example, cell cycle proteins in Alzheimer's disease. So there's this very famous cell cycle reentry hypothesis. And I think our data just provides a new adjustment, a new perspective on how and why Alzheimer's disease brains sometimes show these, for example, cyclin proteins expressed in the neurons. Also, and this is why this little plant image is down there, I think this relates very much to the general biological principle of that damage causes dedifferentiation. So, for example, when you cut a leaf, if you cut an axolotl's limb, for example, the cells that experience the stress, also heart infarction, for example, cells at the border tend to dedifferentiate and try to mitigate the damage either by repopulating and regrowing leaves or limbs, or just by mitigating the damage by transdifferentiating or dedifferentiating into a more favorable environment of the damaged side. And what we think is that something similar is happening here, just over a very prolonged process also in the aging brain that's affected by Alzheimer's disease. And obviously this cell biological relationship to cancer really got us fascinated, because this was something like we can learn a lot from cancer, not just like from the biology of understanding which pathways are involved and how, but also maybe cheat sheet a little bit from the cancer field, because there are just so many compounds, so many treatments developed already that we can maybe use in this context. And one very interesting feature about cancer is that it's not only caused by mutation and driven by mutations, but then the cellular metabolism really plays a key role in providing, for example, the metabolites, or putting these cells in a state to dedifferentiate into cancer cells and then start to proliferate. And this is something that we kind of saw in the IN model in a way. And in this context, neurons are very, very interesting in the context of metabolism. And I mentioned this earlier, neurons are very inflexible when it comes to how they want to produce their energy. They rely on mitochondrial oxidative phosphorylation, but they hate to do anything like glycolysis or to promote, like, for example, proliferation or tissue growth. So they are a very interesting thing to look at. And over the course of the project, we had accumulated a lot of data, transcriptome, DNA methylation, ATAC-seq data, and at the time there were also many omics studies coming out from just post-mortem brains and new data sets from the ROSEMAP study, and so on and so on. And this is what postdoc Larissa did in the lab, to like really overlay all these data sets that we have with the data sets that are out there from postmortem brain, look at the overlap, see which gene modules, so this is just a WGCNA where you can identify modules that are enriched for certain co-regulated genes, and then overlap them with the data sets. And I don't want to go into detail, just showing you this pyramid, and see that on the top of this pyramid we find one gene that seemed to be connected to virtually all of the important pathways that were co-misregulated in our model as well as in the brain, and this is PKM, the pyruvate kinase M. And when we were very excited, looked at our data set again, looked at the differential expression of PKM, and we didn't see a change. This could have been well the stop of the project if there wasn't so much literature about PKM in cancer, while it's virtually like undescribed in like neural development and Alzheimer's disease, for example, and that we learned pretty quickly that PKM is really regulated by splicing. So there's alternative splicing of exon 9 or 10, giving you either PKM1 or PKM2. So PKM1 forms these tetramers, which are catalytically active and do catalytically in metabolism what PKM should do, which is basically promoting that pyruvate to go into the mitochondria to fuel mature oxidative phosphorylation, for example, in a neuron. While PKM2 likes to hang out alone, it likes to form maybe dimers, which are metabolically quite inactive. And so we looked in our cells, and while we didn't see total levels of PKM to change, we saw a quite significant increase of PKM2 versus PKM1, also on splicing side, but also on the protein level, which you see here in the stainings. And then in line with the metabolic feature of these two isoforms, we see that in the ADINs, we see a reduced PKM metabolic activity in these cells. To just do a reality check again, if this is something that's happening in the brain, again, we looked into, in this case, ROSEMAP studies, and you see PKM1 isoform and PKM2 isoform. Sorry, the colors are switched, so here red is green and green is red. Actually start with the Bragg stages already very early on. PKM1 is lost and PKM2 becomes upregulated. However, keep in mind that this is whole tissue, so this could also relate to glial activation. For example, activated microglia would start PKM2 over PKM1, and that's also well described, but just giving us another hint. So we also looked at the slides, at the pathology of these cells, looked in neuron-rich regions, and we do see that total PKM2 protein in neural dense regions in the human cortex is actually upregulated. And when we look closer with nu-N as a neuronal marker, we actually see that the intensity, especially in neuronal nuclei, is upregulated in AD versus controls. It took us quite some convincing of having the pathology people to stain a breast cancer marker in our INs, but after this, it's pretty clear that there's really a strong differences between AD cases and controls. To then really look into the metabolism of these cells, we teamed up with Angelo D'Alessandro at UC Denver, did mass spec metabolomics, and what you see here, you see the gene names from the RNA-seq and the metabolism, that you see a strong increase and enrichment of canonical glycolytic metabolites in the ADINs versus the controls, and that this traffic jam, I don't really have a pointer here, just above PKM2 and PKM1, you see an increase of the metabolites, indicating that this protein is just not, the enzyme is just not as functional, indicating that there's really like a Warburg-like effect, which is the switch from oxidative phosphorylation to aerobic glycolysis, which has been heavily described in cancer, is actually happening in the AD neurons. This is not the only thing that PKM2 does. PKM2 also becomes phosphorylated by, for example, kinases involved in development in cancer, moves through the nuclear pore into the nucleus, there becomes acetylated and interacts with transcription factors such as HIF1 alpha and STAT3, which are very well-known stress and also cancer-related master transcription factors, leading to the expression of stress and pro-apoptotic genes. They establish a glycolytic gene expression, dedifferentiation, and generally oncogenes. So I already said that in the postmortem brain we saw nuclear PKM2, and here I also show you that we see phosphorylated PKM2 enriched in the neuronal nuclei of ADs versus controls, and this actually engages with a chromatin at promoters of HIF1 alpha and STAT3 target genes, making them more accessible and driving that gene expression, and this is in contrast to cancer, actually leads to increased competency for apoptosis in these cells. So usually a mature neuron does not want to do apoptosis, it wants to stay with us from when we are born or before till the end of our lives. So these cells are really resistant to apoptosis. But when these cells dedifferentiate and become more immature and they become more stem celly, they actually can perform apoptosis again, and this is what we show here. So these cells are not dying spontaneously, but if we put pro-apoptotic triggers such as a BCL2 inhibitor in this figure, the ADINs can die overnight, while we think that overall, like in the progression of Alzheimer's disease, these are other signals, for example, buildup of tau or amyloid beta, that over time, years and decades, can really lead to a cell death of neurons, which might be in a programmed way. I'm not saying that it's only apoptosis. So the last thing, 2.5, 3 minutes, that's perfect, is that we can also benefit from the developments in the cancer field and finding certain drugs that go at these targets. And here I will just show some slides on a natural compound, which is chiconin, which has, however, inspired a lot of drug development in the cancer field as in this context of the anti-Warburg, anti-cancer metabolism drug development. So chiconin is not really a very good drug. It has a lot of side effects, but it's a tool compound for us, and it's also a lead structure for development of anti-cancer molecules. And chiconin is known to modulate PKM2. And modulate, I mean that it activates the metabolic activity of the PKM2 isoform to act more like PKM1, maybe by tetramerizing it, but there are also specific compounds that are designed to tetramerize PKM2 to act more like PKM1, but then also to inhibit the translocation into the nucleus of phosphorylated PKM2 and then to shut down the STAT3 and HIF1 alpha related gene expression. So we applied PKM1, first looked at the metabolome of these cells, and what you see here is that this enrichment of glycolytic metabolites is actually almost completely rescued when you treat these cells with chiconin for about a week, which was a very nice thing to see. Also, when we looked at the apoptosis competency, that could be reduced basically to normal levels again. But when we looked at the transcriptome, and here you see just 3 controls and 8 AD patients on a principal component 2-dimensional projection, and yet you see that the controls are residing on the upper left corner while the ADs are more on the lower right corner. And when we apply chiconin, they don't really get exactly like the controls, but they all move into this upper left direction, indicating that on the whole transcriptome, on the whole identity of the cells, what the cells want to do, we're moving them closer to the controls again. However, when we map this again to like how much of the mature identity are we rescuing, it's about 50%. So this is not the final answer, but it's very inspiring to now try to use also more drug-like molecules on these INs to see what they do about these AD phenotypes. So in summary for this, just highlighted that the cancer-like PKM2 isoform causes a fate loss and contributes to the fate loss in Alzheimer's disease neurons, and it has both a pathogenic metabolic loss of function role, but also the effect is strongly mediated by a gain of function epigenetic transcriptional cofactor role in the nucleus. Also, we showed that the neuronal dedifferentiation, which I described, can lead to a cell death competency. This is partially mediated by PKM2 and probably the interaction with HIF1 alpha and specifically STAT3 is important here. And also just like globally note that we see quite many mechanistic similarities on the pathway level of the role of PKM2 between neurons, Alzheimer's disease and cancer, but also quite some significant and distinct differences. For example, if a cell decides or ends up being more competent for apoptosis, while, for example, most tumors in general evade apoptosis by downregulating it. And we don't really know why we see these inverse effects at the moment. And then, of course, since it's a pharmacological conference, that the pharmacological modulation of PKM2 activation and inactivation of the nuclear parts may prevent neuronal AD-related fate loss, and with that might at least slow down or maybe even prevent Alzheimer's disease pathology. So with this, I would like to end and thank everybody who was involved. My lab that just moved from Innsbruck in Austria to San Diego, and I was fortunate to have a long list of people that got worked with me in Innsbruck to move with me. The funding sources, of course, and then thank you for listening. And I think we have like 7 minutes time for questions.
Thank you very much. Thank you very much, Jerome, for the super nice talk. Very interesting. Questions? Daniela?
Hi, Jerome. Thank you for the very nice talk. I have a curiosity question. So it was shown that actually in neurons, they can do the Warburg effect, particularly in high level of activity, so to, in a way, protect themselves from the possible oxidative stress. So I was wondering if you try to challenge your IN to modulate their activity and see how they respond in that sense with the Warburg effect or not.
That's a very good question. So what we think, what's pathological about the things we see here is that they become chronic. And so for acute stress or acute activity, we don't really know, and it's not that easy. So in the cultures, we can use, like, for example, glutamate or so to just like overstimulate the cells, which would be more a model for pathology, I think. But we're working with optogenetics now to kind of fine-tune this and see like how much activity input is good for the cells and how much is too much, or when does it become too chronic. And one thing we definitely want to look at is PKM in this context. So that's a very good suggestion, yeah.
Thank you very much. I have a general question. You mentioned that some of the experiments were in brain slices, but most of the experiments were in these induced neuronal cultures, which I assume lack the natural environment, the other cell types like glial cells and astrocytes and so on. So is it possible to add those cells to these cultures to reproduce a more physiologically relevant environment for the neurons?
Absolutely. Like, this is like a major focus of what we're trying right now. So unfortunately, they're not really good validated protocols to produce through direct conversion aged astrocytes or aged microglia. Astrocytes, there are now some protocols and we're trying them. So we're currently making these cells from iPS cells. So we're having the old neurons with the young, fit baby astrocytes and seeing how they communicate on, for example, the metabolic level. So this is definitely where this will go to. And we don't really know how the astrocytes, for example, would react to all this lactate that's around now. Maybe they would just like buffer this away. If you have like fit baby astrocytes, maybe there's just no phenotype left in the end, and they would like completely buffer this out through lactate uptake and then maybe glutamine exchange. So we don't really know that, but we're very curious to see what the glial cells have to say about the phenotypes of these monocultures of human neurons.
Over here. Yeah. Oh, one last question. Yeah, thank you very much. That was very exciting talk, actually. Powerful technique for ions. My question for you is, did you look at any of the architecture of the neurons when you're looking at talking about immature state in the ADINs? Makes me wonder what's happening at the synaptic level or at dendrite formation and stability.
Yeah, we haven't really looked in detail very much. We've seen that the cells just like on a kind of a 20x reconstruction of morphology show less branching, and they also show less synaptic dots, and with that I mean overlay of PSD95 and synapsin, again with a resolution of like 20 to 40x. So we haven't really done like really high-resolution synaptic structure experiments, but I'm pretty sure that there would be changes there, and it would be interesting, like how also, for example, like a PKM treatment with, for example, chiconin, if it would have an effect on the synapses of these cells.
Yeah, very exciting. There's a lot you can do with your model. Yeah.
Okay, thank you. I think we can move to the next speaker. Thank you, Jerome, again, and invite Daniela Mauceri to give her talk.
Thank you, Elena. And I also would like to thank the organizers for giving us the opportunity to present in this symposium the latest work from our group. So there is no doubt that there is in the nervous system a tight link between structure and function. Evidence comes from the multitude of studies reporting alterations at the level of structural of the synapses. Maybe there are more, maybe there are less. The dendrites are impaired, axons are changed, even the soma shape has been found sometimes to be altered. And this has been shown, as I said, for a multitude of different disorders, whether it's with a genetic component or not. Even just in normal aging, we know that there are structural alterations that are reported in neurons. So the understanding of the molecular and cellular mechanisms that are governing neuronal architecture may have far-reaching implications from a therapeutic perspective, and this is one of the main focus of our group. A little bit more than a decade ago, we were trying to identify which genes under synaptic activity, but also under the control of nuclear calcium, which is one of the main drivers regulating gene expression in neurons, may be critical for shaping the structure of pyramidal neurons at the synapse, but also at the dendritic level. And back then, we were lucky enough to identify one of the prime regulators of dendritic architecture, which is vascular endothelial growth factor D, or VGFD. Like the name suggests, VGFD belongs to the vascular endothelial growth factor family. However, it is encoded by a completely different gene. It has a completely different regulation for its expression, and it also has a completely different biological function, particularly in terms of what it does in neurons. So we showed that VGFD is critical for stabilizing and make sure to tell the neurons to maintain the mature dendritic shape that they have. So in one of the key experiments that we performed, we use recombinant adeno-associated viruses to silence the expression of VGFD, specifically in the CN1 neurons in adult mice. And this was sufficient to trigger a loss of dendritic complexity, so that the dendrites of these neurons were shorter, were also less branched, and overall simplified. As a result of the fact that we reduced VGFD levels and that we had this impairment in the dendritic structure, we also saw that the mice had impairment in their memory formation. In a classic Morris-Water maze test, the mice receiving a scrambled virus remember where the platform was, and they spend considerable more time looking for it in the target quadrant in order to escape the water, because they don't like to swim in general. However, the mice with reduced dendritic carborization in the CA1 neurons, because we reduced VGFD levels, did not remember anymore where the platform was. And this was further confirmed with another hippocampal memory-dependent task in contextual field conditioning. The mice are exposed to an electric foot shock in a particular environment, and when you put the mice back there, you measure the amount of time that they spend freezing as a proxy for the fact that they remember, this is not a good place, I don't want to be here, I'm afraid of it. However, the mice with VGFD reduction and a loss of their dendritic arborization did not remember anymore and spend less time freezing. So in conclusion, also with a series of follow-up studies that we did, we're pretty sure now that we can say that VGFD levels are critical to drive and maintain a particular dendritic tree, which is optimal for that particular neurons, and this is associated also to proper cognitive function. We also did quite a bit of work characterizing what actually ensures the neurons are expressing VGFD. We know that it's under control of basal synaptic activity. The synaptic NMDA receptor is involved. We know that nuclear calcium signaling is involved. And in the last few years, we also showed that the neuronal expression of VGFD is under epigenetic regulation, particular of histone deacetylases. What we did not know yet, and then we started also asking ourselves this question, was whether under toxic or neurodegenerative condition, this gene, so this protein, would be somehow affected. And in order to tackle this question, we started with a very, very easy experimental paradigm, something that we were sure that would trigger the degeneration of neurons and eventually their death. So we used culture primary neurons, which we exposed to a bath application of high concentration of an NMDA, which triggers the activation of synaptic NMDA receptor, but mostly the activation of the extrasynaptic pool of an NMDA receptor, which is one of the prime modulators of toxic signaling, leading then to degeneration and eventually the death of these neurons. And we saw that there was a time-dependent as well as concentration-dependent downregulation of a VGFD expression as a result of the bath application of an NMDA. To confirm that this was indeed mediated by the toxic component of the NMDA receptor pool, we also isolated pharmacologically the extrasynaptic receptors so that we prevented the activation of the synaptic ones, even in the presence of an NMDA. And even in this case, this was sufficient to trigger downregulation of VGFD. Finally, we also tried to promote, so to say, the activation of extrasynaptic NMDA receptor by using the neurons against themselves, so that we were pushing them to have a glutamate accumulation in the synaptic cleft that eventually spills out and would activate these receptors. This is possible to do using a paradigm here, the acronym is OGD, so glucose-oxygen deprivation. Basically, the neurons, they start to not be able to reuptake glutamate anymore, and then this is the result. You will have the activation of these toxic receptors, and also in this case, VGFD levels were downregulated. So then we started asking ourselves, okay, are there then conditions when this is known to happen? So when we know that there is an activation of this toxic signaling, and can we then look at what would happen to VGFD? And one of the best characterized situations in which this may happen is actually stroke. So we established a mouse stroke model, the MCAO, or middle cerebral artery occlusion, in which we cauterized the middle cerebral artery in the mice, which eventually leads in the ipsilateral side of the cauterization to a large infarct area so that you can assess the damage to the brain parenchyma. Now, as expected, because this was already known in the literature, that when you have such stroke events, this eventually leads to a loss of dendritic architecture. There is a structural disintegration of the dendrites of the pyramidal neurons in the motor cortex in layer 2-3. So this, let's say, was not a surprise. We confirmed that we could see this structural disintegration. But then we looked also at VEGF-D levels, and confirming what we saw in vitro, we saw a downregulation of VEGF-D, both at the mRNA as well as the protein level. Now, considering the fact that we knew from our previous work that VEGF-D is really one of the prime mediators for the stabilization of dendritic architecture, and that here we also had this loss of dendrites, this of course started calling us to think about a supplementation strategy, also to see if the two events would be really correlated. Moreover, we are generally using and working under these hypotheses in the lab. This, of course, is an oversimplification of the situation, but let's say that we can consider a normal healthy neuron and its functionality as a result of a series of building blocks that have to perfectly align to make sure that the neuron will work in a certain way. And under any sort of pathology, so when we have and we're looking at damaged or dysfunctional neurons, then the building blocks are thrown off balance. And structural aberrations may not be the cause of the disease, but they are, let's say, one of these building blocks on the road to dysfunctionality. And at the same time, and this is why we are looking at that, they represent a potential point of intervention to try to somehow normalize again the neurons. We are again not aiming to cure the disease, but perhaps it can be somehow beneficial. So we started to design a potential supplementation strategies to replenish VEGF-D levels. So as a first thing, we decided to deliver VEGF-D by stereotaxic injection into the ventricle of mice after the stroke event. VEGF-D, it's a secreted protein, and moreover, I mean, there were already a lot of labs in the world working with it, so this is something that we can just buy, the mature, fully functional VEGF-D, and then just deliver it to the mice. And as a first thing, with morphometric analysis, we could show that in comparison to the control animals that received either the vehicle or an unrelated protein, such as GFP, here the stroke event, as expected, caused a loss of dendrites in the ipsilateral side to the cauterization, but the mice that received one single delivery of VEGF-D showed absolutely no loss of their dendrites. We then look at the infarct size, and also in this case, the single delivery of VEGF-D was sufficient to reduce the infarct size of the mice in comparison to those ones that were just receiving a CSF. And this was specific for VEGF-D because when we treated the mice, for instance, again with the GFP, but even with proteins that are related to VEGF-D, so we supplemented them, for instance, with VEGF, or even VEGF-C, which is the closest homologs in terms of structure and sequence to VEGF-D. None of them protected the brains of the stroke mice from damage. And then finally, we also wanted to have a functional readout, so to show, okay, we have upheld integrity. This is beneficial because at the end we have a less infarct size, is then good for the motor performances of the mice. And here I'm just showing one of these tests. So what happens is that when you have such large infarct area in the mice and you place them in front of a corner with walls of about 30 degrees, the mice always turn, paradoxically, always to the same ipsilateral side of the damage. So that they develop a so-called laterality index. However, when we treated the mice with VEGF-D, this laterality index was not developed anymore. So showing that not only we were preserving the architecture, we were having a reduced brain damage, but also the motor behavior of the mice was somehow normalized. Now, the delivery intra-stereotaxic injections is not something that one can easily envision to do somehow one day in a clinical praxis. So we started to look at alternatives for route of administration to deliver VEGF-D to the brain. And this was the time in which, in the literature, but also there were clinical trials, they were gaining momentum, and they were using the nose-to-brain delivery as a route of administration. So we decided to use this approach, and we developed a way to give a VGFD with the nose drops, again, after the stroke. I mean, of course, we also checked that we had proper delivery so that we could detect it afterwards in the brain of the mice. But critically, we also saw that we had exactly the same results that we could achieve with a direct stereotaxic injection of VGFD protein. So that also in this case, we could preserve the dendrites from the stroke-induced damage. We had a reduction in the infarct size when we were delivering VGFD with the nose-to-brain approach. And also, there was no development anymore of the laterality index, so that again, also in the motor performances, with the delivery via the nose of VGFD protein, we were successful, let's say, in reducing, at least to a certain extent, the damages induced by a stroke. We then decided to take even a step further to optimize, let's say, this approach even more. And for that, we looked at the structure of VGFD, and we focused in particular to this helical portion, which is the one that is known to mediate the interaction and the activation of a VGFD cognate receptor, which is VEGF receptor 3. So we collaborated with a chemist group at Heidelberg University, and we generated a library of VGFD peptide mimetics based on this helical portion, so with the classic alanine scanning, but we also introduced a non-natural amino acid that would either stabilize or destabilize the helix. And then we tested the peptides to select the most potent. So we wanted to find those that would actually be even more potent than VGFD, but at the same time, they would still retain their specificity of activation for this particular receptor. After we managed to do the screening, those ones that passed the selection were then applied with the same approach of the nose-to-brain delivery using the nose drops. And we used these VGFD mimetic peptides that at the time we called FMPs. So here I'm showing data for 2 of these highly potent VGFD mimetics that behave exactly as VGFD, so that with the nose delivery, they successfully prevented the dendritic disintegration that is triggered by the stroke event. They reduced the infarct size, and they also prevented the development of this laterality index with the mice always turning to the damaged side. Now, none of these results were achieved when we used the control peptides, so that we generated scrambled with exactly the same amino acids, just in a different sequence. None of these results were then could be seen, so that we had no protection anymore of the dendrites, no reduced infarct size, and the mice developed, as expected, their laterality index. So in conclusion, for this part, we could then say that not only, let's say, normal VGFD levels are important for maintaining and stabilizing normal dendritic architecture, but when this is somehow compromised, one can think about using a VGFD supplementation strategy to try to prevent structural disintegration and then eventually also preserve functionality. But then we started asking, what if this is, let's say, of course, it was excellent, we were quite happy with the results, but what if this is just applicable to pyramidal neurons, to these particular disorders, to this particular area that we are looking at? So we were looking at the motor cortex, and we cannot apply it to anything else. So we decided to look at a completely different areas and a completely different neuronal population. So we moved to the retina, and we tried to establish the model. Luckily, there was already something in the literature that was, well, let's say, quite easy to then set up in the lab to trigger the degeneration and then eventual death of RGCs, of the retinal ganglion cells. So the retinal ganglion neurons are those neurons that eventually will transmit the visual information from the retina to the brain. And it is well described in the literature that by an intravitreal injection, into the eye of the mice will trigger degeneration of these neurons and eventually their loss. We could definitely confirm that, so that here I'm just showing the data with the staining for this marker, that is BRN3AA. This is used to identify the RGCs, but we also check that before the degeneration, they also started to have a disintegration of their structure. And one can also count in flat hole mount of retinas the actual number of neurons that are still there, and we could definitely see that over time there is a loss of these neurons. We then also confirmed that VGFD is also expressed in the RGCs. So in the mice, we can detect it, but we also collaborated with an eye clinic in Heidelberg, so we were lucky enough to get eye from human donors, and also there we could detect VGFD expression in the RGCs, both at the protein, but also at the mRNA level. And what we saw when we subjected the mice to the NMDA injection was that VGFD levels was going down. So then similar to what we have seen before. So then also in this case, we decided to go for our supplementation strategy. So we injected VGFD in the eye of the mice and then check what would happen. We could see that VGFD protected the structure, but here, again, I'm just showing the cells themselves. And as expected, NMDA causes the loss of these RGCs that was completely abolished by the delivery of VGFD. We also then looked a little bit further down. So I mentioned before that the retinal ganglion cell neurons are those ones that eventually will transmit their information to the brain because their axons builds the optic nerve. So we also looked at the integrity of the optic nerve. Here there is this marker in green which means basically damage, and I guess that even by eye you can see that in the NMDA optic nerve, the NMDA treated optic nerves, you can see that there is an increase of this proxy for the damage, which is reduced in the eye of the mice that received VGFD. We also then tested the functionality, so to make sure that not only we had preserved the structure and the vitality, but that it would still be able to transmit information. So we did pattern electronography, and the red line here is the line from the impaired transmission from the NMDA injected eyes, while those ones that receive VGFD are basically indistinguishable from the controls. Now, something that we came across when we were characterizing the NMDA injections was something that we were somehow not expecting. That is that we started looking also at the vasculature, first to just in general characterize what was going on in the retinas, but also because VGFD, as I said at the beginning, belongs to the family of vascular endothelial growth factor. So we always keep an eye on what is going on in the vasculature. And what we saw was that the capillaries in the retina, which are typically constructed by basal membrane, mostly by collagen 4, and then inside there is the layer of endothelial cells. As a result of the NMDA injection, we had an increased formation of these so-called collapsed empty sleeves that can also be found in the literature as acellular capillaries or string vessels and so on. So for us, this was somehow a bit of an oddity. We realized that we were quite ignorant because when we went then to the literature, we found that this was something that was very well described. Actually, the formation of these empty sleeves can happen in human, in a lot of research models, has been shown in different areas of the CNS, so in the brain, spinal cord, in the eyes, but also in the rest of the bodies. And the first time that it was described, it was actually in the 1838. This is not a typo, it was actually the 1838. So that it was known that this happens, so that there is the formation of these collapsed empty sleeves that are non-perfused anymore, and then eventually will then contribute to the further damage of the surrounding tissue. What we were then even more surprised to see was that when we were doing our supplementation strategy, so when we were targeting and protecting the neurons from degeneration, we also somehow ended up preventing the loss of endothelial cells and the formation of these empty sleeves. Now, from a temporal perspective, when we did all the characterization, we knew that we had first the loss of neurons in comparison to what was going on with the endothelial cells. So we kind of thought, okay, perhaps it's really that the integrity of the neurons is important, also to modulate the integrity of the capillaries, which was somehow counterintuitive, because normally it's always described that it's the vasculature that is important, of course, to make sure that the neurons are okay. And in our model, it somehow seemed that it was going in the other direction. So in order to verify this and understand a little bit better what was going on, we decided to use another approach. So we know that VEGF-D can act on VEGF receptor 3, and this receptor is expressed on both cell populations. So it's expressed on the RGCs and is expressed on the endothelial cells. So we decided to silence the expression of VEGF receptor 3 specifically only in the RGCs. So we made use of viruses that, due to their tropism as well as a combination of promoters, would only be targeting the expression of this receptor in the RGCs and leaving the expression unaltered in the endothelial cells. So that even if we provide VEGF-D, this is still, in principle, still capable to go and act on the receptor on the endothelial cells. And then we did the same experimental paradigm that I described before. So first, in the mice that receive the scrambled virus, as we described before, the NMDA caused the loss of RGCs, the formation of these collapsed empty sleeves, which in both cases could be prevented by the delivery of a VEGF-D. However, when we then silence the expression of VEGF-D receptors, so VEGF receptor 3 in the neurons, we lost the protection provided by VEGF-D, so that the neurons were still losing the dendrites and eventually dying, and also the endothelial cells were doing the same. So suggesting that we really needed to have this signaling of the VEGF-D pathway in the RGCs to drive and maintain the integrity of the neurons, and then eventually this would be beneficial also to prevent further capillary damage and so on. Now, where do we go from here? So in the last years, we actually got to know a lot about how VEGF-D expression is regulated. We know, and we have no doubt, that it's critical for the modulation and stabilization of dendrites, but we are still not entirely sure how VEGF-D does that, and this is where we are now directing our efforts. So what we are doing is that we characterized now the possible effects that VEGF-D has on the different cytoskeleton components. So in order to understand how does VEGF-D does its job, so how does it stabilize its dendrites, we started by looking at dendritic spines. So we looked at F-actin in spines, but over there we see no effect. VEGF-D does not seem to affect spines at all, which confirms something that we already knew from our first studies, that this protein seems to be really key just for the dendrites and not for any other compartment, whether it's the soma or the spines. We then used atomic force microscopy to probe the elasticity of the membrane of neurons and probe the cortical actin, so this network of actin that is just below the membrane of cells. And we saw that VEGF-D actually increased membrane stiffness, so it's a little bit as if it's providing some sort of molecular corset and freezing the neurons in the state that they are by reducing their elasticity when they get poked. And then finally, of course, we looked at the prime cytoskeleton component of dendrites, which are the microtubules, and we found that VEGF-D triggers the stabilization and the lifetime of microtubules. So we kind of know now that this is the pathway that is most likely affected. Nevertheless, VEGF-D, secreted protein, VEGF receptor is activated, eventually impinges on the cytoskeleton to then mediate neuronal structure, but there are still quite a little bit of boxes that we are trying to fill to really define what are the proteins and pathways that may be involved there, with always the idea that eventually they may be representative of future druggable targets one day. So we used two different approaches. So on one side, we did a cytoskeleton-related phosphoarray to identify those modulators of the cytoskeleton that would be under the control of VEGF-D. And on the other hand, we are now dealing with the VEGFR3 interactome by immunoprecipitating the receptor followed by mass spec to identify their binding partner. And I hope in the future to be able then to fill these boxes. Now with this, I would like to conclude. I would like to give thanks to the people in my group, particularly Annabelle Schluter, who is now left. She did all the retina work. Bahar Aksan, who is doing now with everything that is VEGF-D related, the past and present collaborators, the source of funding, and of course, you for your attention. Thank you.
Can I ask you, thanks for your talk. I was going through your paper, the PNAS 2020, and about the intranasal delivery. So I wonder first if you're still pursuing this strategy, and second, I read that you weren't able to identify the recombinant DAGFD after injection. So I wonder whether you have solved this problem, whether you have more data about the distribution of your drug, of your peptide.
So that's, thank you for the question. The problem that we somehow had for the detection, I mean, we also tried, so we were helped also from a series of local, we tested different local companies that would try to assist us with the detection of VEGF-D. The problem that we had is that VEGF-D is also the endogenous protein, so it makes it very difficult then to distinguish what was the one that we were delivering from the one that was already present. In Heidelberg, at the moment, it was basically impossible to get from the animal research right to do radioactive targeting. That would have been the best. This is actually what other people have been doing for the delivery of small proteins and so on. So what we know is that because we know a little bit of what VEGF-D activates downstream, so there are certain kinases that we know are under that control, and we saw by isolating the different areas that there was a activation of these particular kinases, so that we had like a biological readout that we had an increase of these signalings, and we saw really a gradient, and it was also time dependent, so that at the beginning we saw it mostly in the olfactory bulb, but then it started to be detectable also in the prefrontal cortex and motor cortex and so on. So it was a little bit of a gradient. What we did was with other proteins that were maybe a little bit easier to identify, so we could see GFP, and then you really see it in the cells. Or, for instance, we also found a recombinant version of the CRE recombinase, and then we were using a mouse that had Mauceri under the control, so the floxed Mauceri, and then again you can see that there is the formation of the red cells as a gradient in that. Whether we are pursuing that or not will depend again, sadly, a little bit about the animal research thing. So the plan is yes, that we would like to keep that, also because it is quite easy to do, it's really not invasive, and it worked quite well. But so the plan is yes, let's see.
That was a really nice presentation. It
Thank
really
you.
was. So congratulations. In fact, I was in the big IMI NewMeds collaboration, which determined that effectively all schizophrenia is probably this genetically modified is due to changes in neuronal plasticity. And so I've worked on FGF, BGF, most of the neurotrophic factors. I haven't worked on this one. And so what I would like to get a feel of is how you relate that to, say, an activity-dependent neurofactor like BDNF in terms of its spectrum. And that's an issue I would really like to to see, to put it in the panoply of protective factors?
Thank you for the question. So, well, so BDNF, of course, is also key for, particularly for dendritic architecture, although it is a little bit more prominent during developmental stages. When the neurons are mature, then BDNF then takes a bit more role, maybe in the synapses and so on, not so much, let's say, in the maintenance of the mature neurons. So there we see, at least in some experiments that we were doing, it seems that VGFD seems to be a little bit more prominent. Now, what is also a little bit interesting is that we know that BDNF can also trigger the increase of action potential bursting, so it can modulate synaptic activity, and that actually seems to have an effect on VGFD expression, so that there seems to be also a link in that regard, so mostly at the expression level.
Really nice.
Yeah.
Thank you. Last question, because we are a little bit late.
Thank you for a beautiful talk. I just was wondering, particularly in your stroke work, was there any effect on the vasculature?
So we checked for that. I mean, it was, of course, one of the questions of the reviewer. We got used to it, to the fact that when you deal with VEGF, this is the question that comes. And we did not see anything. So we checked for angiogenesis, all sorts of vascularization. We didn't see much. We could not really check these empty sleeves and so on, because the damage is a little bit too much. So the stainings were always not working that much. So we were checking in the peri-infarct area, and there we didn't see anything. But within the infarct size, I cannot really answer the question. So there is definitely no angiogenesis, though, which is one of the things that actually people were expecting.
Okay, thank you, Daniela, and I invite Roberta Facchinetti for her oral communication.
Okay, good morning, everyone. I want to thank the organizers for inviting me. It is really an honor for me to have the opportunity to share my results here with you in this symposium. First of all, just to give you a recap, in addition to neurons, the central nervous system also comprehends glial cells. And the most numerous of them are astrocytes that, as you can see here in this slide, control so many brain functions at so many levels of organizations that are considered as guardians of the central nervous system. Among their innumerable functions, astrocytes support the proliferation and maturation of oligodendrocyte precursor cells into mature oligodendrocytes that are the glial cells responsible for the saltatory rapid conduction of the nerve impulse. This process of differentiation foresees the passage through different stages of differentiation, each of them characterized by the expression of specific markers. For example, the transcriptional factor OLIC2, which is expressed throughout the whole process of differentiation, and the myelin basic protein, MBP, which is only expressed by mature and myelinating oligodendrocytes. Physiologically, astrocytes support oligodendrocytes by making direct cell contacts with them, but also by releasing a variety of mediators, such as the transforming growth factor beta, the fibroblast growth factor beta, the fibroblast growth factor 2, and some pro-inflammatory cytokines that are relevant for their differentiation. When the central nervous system is damaged, however, astrocytes become reactive with the reparative intent to restore the lost homeostasis. But if this process proceeds over time, these cells lose the ability to perform some of their homeostatic functions, including supporting oligodendrocyte, of course. The impairment of this astrocyte oligodendrocyte crosstalk has been observed in different conditions, such as the vanishing white matter, Alexander disease, the Aicardi-Gouttier syndrome, which are all canonical white matter disorders. Alzheimer's disease instead has always been recognized as a gray matter disease, but very recent neuroimaging techniques have revealed the presence of structural abnormalities also in the white matter, and they have suggested also the deposition of beta amyloid as a possible driver or consequence of myelin damage. Moreover, the astrocyte-oligodendrocyte crosstalk in Alzheimer's disease has not been investigated yet. Therefore, to try to fill this gap, I isolated primary and astrocyte and oligodendrocyte precursor cells from newborn rats, and I plated them in a transwell co-culture system in the appropriate media, in which they were able to exchange mediators, but not to be physically in contact because of the presence of a semi-porous membrane. In particular, for oligodendrocyte precursor cells, I use a medium which enabled their differentiation. After 3 days of co-culturing, I treated the lower compartment, that containing astrocytes, with the fibrillary form of beta amyloid. In the recent years, I actively contributed to demonstrate the ability of a compound named palmitoylethanolamide alone or micronized with the antioxidant flavonoid luteolin or Co-ultrmicronized to modulate astrocyte reactivity and neuroinflammation in several preclinical models of Alzheimer's disease. The vast majority of these actions were dependent on the activation of the peroxisome proliferator-activated receptor-α, although several mechanisms have been observed. Of note, PA-LUT, it is also able to accelerate in vitro the maturation of oligodendrocyte precursor cells into mature oligodendrocytes. Therefore, additionally to the fibrillary form of beta amyloid, I decided to add in my co-culture system also a PPAR alpha antagonist and a Co-ultra-PA-LUT, of course. And after 48 hours of culturing of treatment, I perform my molecular analysis separately on astrocytes and on oligodendrocytes. For astrocytes, initially I confirmed the presence of astrocyte reactivity as evidenced by the increased expression of the most widely used markers of astrocyte reactivity, for example, the glial fibrillary acidic protein, a cytoskeletal protein that you can also see in the immunofluorescence images, or the glutamine synthetase, which converts glutamate to glutamine, or the high motility group POX1. A nuclear DNA binding protein. Then I went to examine the aforementioned mediators released by astrocytes to support the differentiation of oligodendrocytes, and I found that beta amyloid alters the levels, the transcriptional levels of interleukin 6, interleukin 1 beta, the fibroblast growth factor 2, and the transforming growth factor beta in different ways. The compound, the ultramicronized compound, prevented these effects in a way dependent on the PPAR alpha receptor only for the pro-inflammatory cytokines. Then I went to study oligodendrocytes in my co-culture system, in particular their maturation. And as you can see here, to study their maturation, I analyzed the ratio between the MVP positive cells and the OLIC2 positive cells that they are represented in the images respectively in red and in green. Very surprisingly, the beta amyloid challenge, the toxic challenge, caused an increased maturation of these cells respect to control without altering their maturation, as you can see from the OLIC2 graph. And the co-ultramicronized ultra-PA-LUT compound was able to prevent this maturation. Of course, this counterintuitive result prompted me to study the morphology of co-cultured oligodendrocytes. And as you can see clearly here from these images of MBP positive cells, so mature oligodendrocytes, beta amyloid caused severe morphological changes in these cells compared to the control group. And again, in this time, co-ultrapalute was able to prevent such alterations. Arborizations of all oligodendrocytes or their complexity is strictly related to their ability to myelinate axons of neurons. Therefore, I deepened my analysis by performing a shoal analysis on MBP positive cells, so mature oligodendrocytes, and I found that the challenge with beta amyloid significantly decreased the number of intersections at 10, 15, and 20 microns from the soma respect to control cells. And again, co-ultrapalute compound was able to prevent such alterations with a mechanism, in this case, dependent on the PPAR alpha receptor. Moreover, similar overlapping results were obtained when I counted the maximum number of intersections, their sum, or their mean of intersections. In summary, with these in vitro results, we provided new findings in the astroglial response in Alzheimer's disease. In particular, we found that beta amyloid hampers the proper communication between astrocytes and oligodendrocytes, in particular by altering their maturation and inducing severe morphological changes in mature oligodendrocytes. Moreover, we found a new and never explored ability of this compound, palute, which is safe and approved for the human use, to counteract the negative effects of beta amyloid on oligodendrocyte homeostasis. Now, the next step of this work will be aimed to assess if it is the vulnerability of astrocytes to beta amyloid to hamper oligodendrocyte homeostasis. And so we are working on this. I want to thank all the collaborators to this work, in particular Professor Caterina Scuderi, Marta Valenza, and Epitech Group, who supported me, and all of you for your very kind attention. Thank you.
Thank you, Roberta. Questions from the audience? Alerie?
Nice talk. I was just wondering if you look at whether different myelin proteins change when you expose them to astrocytes and then with your compound. And also whether you have looked at, you see the same changes alone when you treat them OPCs, because they are in the co-culture, but at the end both of them are exposed and have the receptors. So.
Yes, of course. Of course, we are starting to look at the protein-lipid protein, for example, as other markers, but we also want to study not only the mature stage, but also using markers to stain the intermediate stages of differentiation. As for the second question, we have preliminary results, because this is our first aim, in which we challenged oligodendrocytes directly without the presence of astrocytes. And in our conditions, they don't answer to the beta amyloid challenge, at least for the fibrillary form of beta amyloid. So we are thinking about like an astrocyte vulnerability that affects oligodendrocyte homeostasis, but they are only preliminary data, so we are going ahead now. Thank you for the question.
Thank you. I have only a curiosity. Have you also tested other concentration of a beta or a beta oligomers instead of fibrils?
Yes, thank you. We tested several concentrations of both fibrillary and oligomeric beta amyloid. In our conditions, we found that the fibrillary form of beta amyloid was able to make our astrocytes more reactive than the oligomeric one at the 1 micromolar concentration. We also saw like a proliferative effect of the 10, for example, the 10 micromolar concentration. I know that the oligomeric form of beta amyloid is able to affect the homeostasis of oligodendrocytes, but now we wanted just to know, just to study the activity of astrocytes indirectly to affect oligodendrocytes. So we decided for these reasons to use the fibrillary form.
Okay.
Thank you.
Thank you. Thank you, Roberta. I think we can move to next speaker. So, Alerie Guzman.
Good morning, everyone, and thanks, Helena, for the invitation to present my work here. For most of my talk, I'll focus on myelin and the interaction between myelin and the immune system. And why do I want to focus on myelin? Well, myelin is one of the most neuroprotective aspects that we currently have inside the mammal brain, and other animals as well. So here, myelin is a lipid-rich bilayer multilamellar layer formed by oligodendrocytes in the central nervous system and Schwann cells in the peripheral nervous system. They basically extend their membrane processes to wrap the axon and provide a protected layer that will prevent axons from vulnerability. In the central nervous system, myelin is formed after birth and is formed by a population of stem cells that are scattered throughout the brain and are kept throughout adulthood, which are known as oligodendrocyte progenitor cells, or OPCs. As you have heard before from Roberta, these OPCs are able to basically reprogram themselves, change morphology, and acquire the expression of myelin proteins that will eventually wrap myelin around the axons. Myelin has a key neuroprotective role, but it's also essential, as you have heard before, to have a fast saltatory conduction of action potential. It acts as an insulating layer that allows action potentials to only be released in the internodal spaces. And this has been a key aspect for evolution, because basically it has allowed to speed up nerve conduction exponentially, as you can see in the graph here, where myelinated axons basically have a much faster conduction of action potentials. However, it is not only a protective layer, it also has an active metabolic role, and it has been shown by numerous groups that is key to maintain trophic support for the axons, because it's not only a membrane-bound wrapping of the axon, but it also keeps cytoplasmic in the spaces in between where they synthesize different metabolic compounds, such as pyruvate or lactate, which then get transferred to the axon to secure axonal survival and growth. Unfortunately, it is now well known that myelin is not maintained across different diseases. For a number of years, myelin has only been studied in the context of primary demyelinating diseases, such as adrenoleukodystrophies or multiple sclerosis. However, now it's well accepted that we do see myelin alterations across the board in several neurological disorders, including primary neurodegenerative diseases, such as Alzheimer's disease, Parkinson, or even psychiatric disorder like autism spectrum disorder. So it's probably one of the hallmarks across the board that we can see myelin alterations throughout. Therefore, it's really important that we do understand how myelin can be formed, and in these cases can be restored once it's lost to try to keep axonal health and survival. Unfortunately, as you all know, with evolutions, humans have acquired higher cognitive functions, but in exchange, they have lost significant regenerative capacity when we compare humans to like lower order animals such as axolotl or salamander. And this is particularly true for the central nervous system, which is known to have a very poor regenerative capacity. However, this is only half of a truth, because unlike neurons, which are very poorly regenerate, myelin has a very significant regenerative capacity. In a process known as remyelination, OPCs that are a pool of adult stem cells that are kept throughout adulthood, as I mentioned, are able to restore the lost myelin. And here you just have a nice picture where you have basically a transversal cut of a healthy white matter, where you have the axons in white surrounded by myelin in blue, which can be lost with either in diseases, or also here is a toxic model that mimics demyelination in the lab. But then this keeps the axonal structure as long as myelin is restored back in a timely manner in this process known as remyelination, which you can see here. And this is particularly relevant for a disease like multiple sclerosis, which I will focus my attention throughout the talk today. Multiple sclerosis is the most common immune-mediated neurodegenerative disease. It affects about 2.8 million people worldwide, and it's often considered the disease of the 1,000 faces because depending on the area of the brain that is affected, it will lead to a high numbers of diverse symptoms. And here in the bottom part, you just have a classic progression of how multiple sclerosis evolves with time. At the beginning, most patients present what is known as relapse-remitting MS, which is characterized by an inflammatory attack mediated by T cells, which basically target myelin as a foreign antigen, demyelinate, lead to disability, which then eventually gets recovered in remission due to remyelination. And this just basically keeps going throughout. However, as disease progresses, about 80% of the patients move to a phase that is known as secondary progressive. And in this phase, they basically are in a continuous inflammatory state, which is mainly located at the CNS, and there's no remyelination, there's no remission of the attack, which leads to an irreversible accumulation of disability. And about 10% of the patients present with a different course of disease, which is called primary progressive, which unfortunately starts already with a progression from the very early stages and has no remyelination happening effectively. So how can we relate these stages to the success or the failure of remyelination? Here you just have a very simplistic scheme depicting the central nervous system where you have axons in gray as well as oligodendrocytes forming myelin around. Then in MS, the T cells, as I mentioned, recognizes myelin as a foreign body, targets myelin, kills the oligodendrocytes, and leaves the axon vulnerable to degeneration. But as I mentioned, myelin has a remarkable regenerative capacity. OPCs are able to respond to myelin damage, and they quickly differentiate into new myelin-forming oligodendrocytes. They restore function and leads to functional recovery. And this can relate to the early stages that I just mentioned, to the relapse-remitting phases of MS, where there's a very tight balance between inflammation and regeneration. However, as multiple sclerosis progresses, and especially when aging kicks in, this process is significantly impaired. Remyelination does not happen efficiently, inflammation outweighs the ability to regenerate myelin, and the axon is left vulnerable to degeneration, and it basically is linked to periods of irreversible disability for patients. I just mentioned remyelination quite quickly, but just to give you a more precise biological timeline of how remyelination happens. Here you just have a schematic view of the central nervous system with axons in blue, oligodendrocytes in yellow, and then other glial cells such as microglia, astrocytes, and OPCs. When the immune system targets myelin, oligodendrocytes die, myelin debris is released, which activates microglia. Microglia phagocytes myelin, release factors together with astrocytes that activate the quiescent pool of stem cells, or OPCs, which eventually get recruited to the damaged area and then differentiate into myelin-forming oligodendrocytes. Several labs across the world are trying to understand what fails in MS and in other neurodegenerative diseases to not be able to restore myelin, and it's commonly accepted that there's two main bottlenecks to this process. One is the ability of progenitors to proliferate and fill up the gap, and the second one is their ability to differentiate into myelin-forming oligodendrocytes. About 60% of MS patients have enough OPCs in their lesions, but they don't differentiate into myelin-forming oligodendrocytes. So for the sake of the talk today, I want to focus on T cells, which for a number of years have been always considered, between brackets, the bad guys in multiple sclerosis, because they are the ones that recognize myelin as foreign, and they're the ones that triggers the disease. However, like everything in biology, nothing is black and white, and T cells are also necessary for remyelination. Work that was published about 20 years ago using a toxin-induced demyelinating model that I will repeatedly mention across the talk, which is based on a lysolecithin injection in the ventral white matter of the spinal cord, which basically kills oligodendrocytes and causes demyelination, which spontaneously remyelinates. Basically, they did this model in 3 different mouse models that didn't have immune cells, or they didn't have CD4 T cells. And what they saw, what you can see here, is that in the lesion, which is always in the upper part of the picture, in the control you have a nice remyelination, which is depicted by the increased number of black rings, which is the restored myelin. And this one is significantly impaired when we don't have immune cells, and especially when we don't have CD4 T cells, suggesting that we do need CD4 T cells to be able to remyelinate properly. But as you all probably know, CD4 T cells are a complex family with different number of subtypes, which have different functions depending on their cytokines and transcription factors. And that's also the key for remyelination. So for example, Th17 cells are known to be negative for remyelination. They do impair remyelination, while regulatory T cells have a positive role in remyelination. And today I'm going to focus on regulatory T cells and how aging affects their role in remyelination. So work that was published in the lab before I joined basically showed, to cut a long story short, that regulatory T cells or Tregs can secrete factors that enhance the production of myelin protein by oligodendrocytes in vitro. And then when a model in which Tregs could be depleted endogenously and then perform demyelination, they observed that when we don't have Tregs, this remyelination was significantly impaired. So part of the work I'm going to show you today focuses on trying to understand how aging might actually alter this regenerative capacity of Tregs in the context of myelin regeneration. So basically, it has been shown by a number of groups, including us, that with aging, we have more Tregs. But as I mentioned before, remyelination is significantly impaired, even if we have more Tregs. Here you just have 2 rodents, one young and one aged, that were demyelinated using lysolecithin and look at the same time point. And as you can see, the aged one has virtually no myelin restored. So this begs the question whether, well, why is this, and which role do aged Tregs play in this impairment? So there's not much literature about the regenerative role of Tregs in general, and there's even less about their role in aging. But one thing that was shown is that they have an impaired migration to the lesion area in the case of muscle repair. And when you restore Treg migration, old muscle repairs more efficiently. So we first look at whether we have different numbers of Tregs in our damaged area where we have no myelin. And we didn't see any difference neither in the number of CD4 T cells nor in the number of Tregs at this time point. It's possible that there are differences at other time points that we are now exploring. So we thought that maybe migration is not impaired in this case. So next we thought that it's plausible that we all know that the CNS is refractory to regeneration, and it's actually quite a hostile environment for any repair process. So we thought that maybe what we could do is increase the number of our Tregs to try to see if by getting more of them, we could actually restore impaired remyelination. So we used an IL-2 anti-IL-2 injection to expand Tregs, which were significantly expanded, as you can see here. But then when we look at the number of oligodendrocytes that we could see in an aged CNS, after damage, it was the same. We didn't see a restored remyelination. So that basically makes us think that Tregs might be actually impaired with aging. And when we look at Treg function, usually their immune functions is quite well kept. But again, there's not much known about how aging might affect their regenerative capacities. So what we did, we basically went back to our in vitro model in which we culture OPCs and then exposed these OPCs to either young or aged Tregs directly isolated from the animal and not stimulated. And what we observed that when we expose OPCs to young Tregs, we have an enhanced production of myelin proteins, CMP and MVP. But this was not the case with the aged Tregs, suggesting that aged Tregs might indeed have an impaired ability to boost myelination. This was also validated in a more complex system using brain slices, where we saw the same thing. Young Tregs wouldn't have myelin production, but aged Tregs would not. And this was quite important because from the publication of the paper in 2017, a lot of trials have been focused on trying to expand Tregs for MS patients, hoping that they would boost remyelination. But this might actually suggest that that might only be efficient as a therapy for a very narrow window of time, since with age, the ability of Treg might be hampered. So we next wondered whether we could rejuvenate these Tregs and restore their ability. To do so, we basically used, this is a bit of a complex slide, but basically we used a model in which we could deplete endogenous Tregs using diphtheria toxin in the FOXP3-DTR mice. And then we could inject either young or aged Tregs that were resistant to diphtheria toxin, and then basically evaluate whether remyelination was happening efficiently, and whether the young receptors, the FOXP3-DTRs, would basically restore the ability of aged Tregs to generate myelin. So here you just have a quantification using 2 markers that are known to be markers of oligodendrocytes, which are CC1 and ASPA. And what you can see in the graph is that when we deplete endogenous Tregs, we do have an impaired oligodendrocyte differentiation, which is a surrogate measure for remyelination. When we then inject young Tregs, we restore this as expected because we are reaching baseline level. But surprisingly, when we inject our H-Tregs, we also do rescue this effect, suggesting that in a young environment, H-Tregs can be restored. So we next obviously asked what was different between young and H-Tregs, and we performed bulk RNA-seq. And basically, to cut a long story short, we found about 1,700 genes that were differentially expressed, 300 of which were downregulated with aging, and most of them were associated, as expected, with immune functions when we look at the gene ontology terms. So with this approach, we actually wanted to identify potential mechanisms that might be altered with aging that could be used as therapeutic targets to boost remyelination in an age context. To do so, we went back to our in vitro cultures because one thing that we had seen is that Tregs were always located in close proximity in these co-culture experiments to our OPCs. So we wondered whether the effect of Tregs, at least in vitro, is soluble or contact mediated, and this could actually help us narrowing down potential candidates. So similar to what Roberta did, we did a transwell assay using OPCs and Tregs. And what we observed was that young Tregs were only able to drive OPC differentiation and MVP expression when they were in direct contact, but not in a transwell, suggesting that at least in vitro, they do need to be in close contact. Then we used this more as an approach to narrow down and find targets that could involve in this process. And what we did was to combine our RNA-seq data, where we basically selected the proteins that were downregulated with aging and expressed in the plasma membrane with an already published database. We came across about 20 candidates, roughly, which we then narrowed down to 7 and perform another in vitro experiment in which we culture OPCs with young Tregs, but previously we blocked these proteins in the membrane with an antibody. And then we evaluated whether once these targets were blocked, Tregs were still able to drive OPC differentiation. And basically here I just show you the 2 positive results that we got, which are with MCAN and integrin alpha-2. Basically, when we blocked MCAN or integrin alpha-2 in young Tregs and then put them in contact with OPCs, as you can see here, they were not able to drive OPC differentiation. So this data suggests that Tregs need the expression of MCAN and integrin alpha-2 to drive OPC differentiation, whether they drive OPC differentiation directly or via activating something else in the T cell when they bind to OPC. That's something that we are currently investigating. And basically just before finishing, I wanted to say that basically the reason why we wanted to look at what was different is to try to find targets that would basically allow us to modulate or mimic what Tregs do to OPCs, because obviously expanding Tregs on top of being limited by the age is also something that is not that feasible because you would cause some other issues. You could basically let them more susceptible to infections and so on. But I thought that this was a good opportunity also to discuss or relate something that has been published by a collaborator from ours, Professor Adrian Liston from the University of Cambridge, where they have actually developed a viral-based therapy to harness the anti-inflammatory role of Tregs in different diseases, including traumatic brain injury, MS, or aging-related cognitive decline. So what they have done here was basically develop a virus in which they express interleukin-2, which is a survival factor for Tregs, only under the GFAP promoter and therefore exclusively in the brain. And with that, they evaluated whether that would improve the outcome of traumatic brain injury, MS, or aging, which they successfully did, suggesting that we could not only harness Tregs using specific mechanisms like integrin alpha-2 or MCAN, which we have described, but potentially also use this technology to improve remyelination or myelin generation in different models, not only MS, but potentially Alzheimer's disease and so on. So with this, I just would like to go through the conclusions. I hope I have convinced you that with aging, Tregs have an impaired regenerative capacity and they do not have the same ability to restore myelin. However, we can reverse this problem by putting them in a young microenvironment. I have shown you that by default, aged Tregs have a significantly altered Treg transcriptome, including inflammatory functions as well as neurotransmitter secretor and so on, and that part of their regenerative capacity is mediated by direct contact between Tregs expressing either integrin alpha-2 or MCAN and OPCs, and that when we block one of those two, basically they are not able to drive OPC differentiation in vitro. And they might be potential new targets to boost myelin regeneration in the HTNS. With this, I would like to thank my previous lab in Belfast, where most of this work was done, my current lab in Alicante, all my funding, my collaborators for this project in particular, and my funding, and all of you for your attention. So I'm happy to take any questions.
Thank you, Alerie. Very nice talk. Any question? I have a question. Do you know whether there are alterations of MCAM or of the other protein that you find in Treg that have been detected in multiple sclerosis patients or in age individuals compared to young?
I don't know in age individuals compared to young, but there's work from Alexander Pratt in Canada where they saw that there's an increase of MCAM in a subset of Tregs with in multiple sclerosis models, and that they are associated to a different clinical score. But they relate that more to their immune functions because they have different inflammatory properties rather than whether they have a different role in remyelination.
Okay, thank you. Okay, if there are no other questions, I think we can move on. And I'm the next speaker. Okay, thank you very much. I'm happy to be here to share with you some of the results we obtained in our lab in the last few years. Our main goal was to find out a molecular crosstalk between the primary mechanism of Alzheimer's disease pathogenesis and synaptic failure. And as you know, Alzheimer's disease is the most common form of dementia, and it's characterized by two main hallmarks. So the senile plaques constituted of amyloid beta peptide and the neurofibrillary tangles of tau protein in the hyperphosphorylated form. Oh, sorry. And already in 1992, Hardy and Diggins posited the amyloid hypothesis according to which A-beta precipitates to form plaques, and then this in turn causes neurofibrillary tangles, and A-beta is able to trigger neurotoxicity pathway, inflammation, and overall, all these signaling pathways lead to neurodegeneration. Therefore, it has been described how A-beta is produced by the cell in details. So we know that A-beta derives from this large protein called APP, amyloid precursor protein, that can be cleaved by two enzymes, beta secretase base 1 and gamma secretase, that with their concerted action release a beta sequence from APP. On the other hand, the cells have another option. There is a non-amyloidogenic pathway, and the main protagonist is an enzyme called ADAM10 that is responsible for the alpha secretase activity, because ADAM10 can cleave APP within the sequence corresponding to A-beta, and therefore it prevents amyloid beta formation. And in light of the importance of amyloid beta peptide in the cascade of events that leads to dementia, in the last 25 years, most of the therapeutic strategies that have been developed were focusing on A-beta peptide. So at the beginning, several molecules, inhibitors of beta secretase base 1 and gamma secretase were designed and tested in order to inhibit amyloid generation. Then another strategy that takes advantage of the immune system was tested. So several antibodies targeting F-beta have been developed in order to reduce the beta level, to increase the clearance of the peptide, and to remove the burden of amyloid in the brain of the patients. And this kind of immunotherapeutic approach has been now also developed for tau protein. However, all these therapeutic approaches, so inhibitors of the secretase, antibodies, were tested in clinical trials, but the results were quite disappointing because they were able, yes, to remove a beta, but the effect on the cognitive function of the patient was not so clear. So in this cemetery of therapeutic strategies, only 2 antibodies survived. So they are aducanumab and lecanemab that the Food and Drug Administration in the States approved via the accelerated approval pathway in 2021 and 2023. And they are recommended for the treatment of Alzheimer's disease patients in the early stages of the disease. However, this approval was, let's say, preceded by a really heated debate because there are several concerns regarding the cost of the therapy, also the adverse events that are due to a beta immunotherapy, but mainly because it was not clear whether they are effective in changing the disease trajectory. So I reported here the results of lecanemab published this year, and as you can see, the effect on the amyloid burden is convincing, so you have a strong decrease of amyloid plaques in the brain of the patients. But if we look at the cognitive function, you can see that lecanemab treatment results in a moderate less decline on measures of cognition and function compared to placebo. Therefore, this question a little bit also the amyloid hypothesis stated by R.D. Diggins. So Alzheimer's disease is not this simple linear pathway starting from amyloid peptide and arriving to dementia. So amyloid beta peptide is correlated with Alzheimer's disease, is central in the pathophysiology, but amyloid beta alone is not sufficient to cause Alzheimer's disease. Further complexity is present in the pathogenesis of this form of dementia. And in this framework, we thought that maybe it was important to study the mechanism underlying Alzheimer's disease pathogenesis in the place where the disease begins. And this place is the synapse. Because if you look at the patients, you can clearly see that cognitive decline correlates very well with the synaptic loss, more than with plaque deposition or tangle formation. And several studies also demonstrated that oligomers of A-beta are able to bind the mature excitatory synapse, and they completely impair the biological mechanism responsible for memory formation and learning, so the long-term potentiation, for example. And in this frame, we decided anyway to change a little bit the perspective. So we were not interested in understanding what A-beta does to the synapse, but we were wondering whether the synapses can be the place where A-beta is generated. So we isolated from the excitatory synapses this, let's say, really complex compartment that is called PSD, postsynaptic density, is an electron zone present in the postsynaptic compartment, and there you can find the glutamate receptor, the signaling molecules, the scaffolding protein, but in the PSD you can also find APP, base, ADAM10, so all the machinery that you need to produce amyloid beta peptide. But in this frame, we decided to focus our attention on ADAM10 because we are pharmacologists and we think that ADAM10 is a good pharmacological target because the upregulation of ADAM10 not only prevents amyloid beta peptide formation, but also increases the production of this fragment, SAPP alpha, that has been shown to be neuroprotective and potentiating synaptic efficacy. But what's ADAM10? ADAM10 is a member of this disintegrin and metalloprotease family, so it's a component of the excitatory synapse, but it's not just alpha secretase. It's not just the enzyme that prevents amyloid beta peptide. ADAM10 is a neuronal sheddase, so it's an enzyme able to cleave adhesion molecules in the synapse, and therefore is able to change the structure and the shape of the synapses. For example, if you mutate in encadering, that is one of the substrates of ADAM10, if you mutate the ADAM10 cleavage site in encadering, you can detect a strong enlargement of the synaptic head. And this is important because the structure of the synapses is directly correlated with its function and the synaptic transmission. And to support the relevance of ADAM10 in the synaptic scenario, we demonstrated that ADAM10 activity is tightly regulated by activity-dependent synaptic plasticity phenomena, because ADAM10 cleaves its substrate only when it is properly localized in the synaptic membranes. So we demonstrated that ADAM10 is delivered to the synapse by the binding of its cytoplasmic tail to SAP97. On the other hand, ADAM10 removal, so the decrease of its activity, is triggered by the association of AP2, that is a clathrin adaptor protein that triggers the endocytosis of the enzyme. And the association to this binding partner, and thereby the localization of the enzyme and its activity, are under the control of LTP and LTD. But what about Alzheimer's disease? So in Alzheimer's disease patients, we analyze the synaptic localization of the enzyme. We analyze the postsynaptic compartment obtained from hippocampal specimen of Alzheimer's disease patients in the early stages of the disease. And we found a significant reduction of ADAM10 synaptic availability. And also in the patient, we measure a significant reduction of SAPP-alpha, that is the main product of ADAM10 activity on APP. And so this demonstrates that in Alzheimer's disease patient, we have a decrease in ADAM10 in the synapse, so in the hippocampal synapse, a decrease in its activity, like if this is a synaptic feature of the disorder. But so we are, again, pharmacologists, so the idea is we found this pathological feature, so how we can tackle this, how we can restore ADAM10 synaptic availability to have a functional synapse. And we continue our characterization of the mechanism that control ADAM10 localization in the Alzheimer's disease patients, and we found both in Alzheimer's disease patient, but also in Alzheimer's disease mouse model in APP-PS1 mice, that there is an increased association of ADAM10 with AP2, so with the clathrin adaptor complex that triggers ADAM10 endocytosis. So in the patients and in animals at the early stages of the disease, at the age of 6 months, we have this pathological alteration of ADAM10 binding to AP2. So to address this issue and to tackle this interaction, we design a tool. We design a cell-permeable peptide in collaboration with Daniela Di Marino at the University of Ancona. And this peptide is constituted by the TAT sequence that is enriched in positive charge amino acid, so it crosses the membranes, and the sequence of ADAM10 responsible for the binding with AP2. So the peptide is able to enter into the cells and to compete with the ADAM10-AP2 interaction, and therefore to decrease the association of ADAM10 and to decrease the endocytosis of the enzyme. We tested several peptides. We have always used as a control an inactive peptide. And at the end, it turned out that PEP3, what we call PEP3, was the peptide more effective in disrupting ADAM10-AP2 association and in increasing ADAM10 postsynaptic levels. So we tested the peptide in several in vitro models, hippocampal cultures, in slices, and then we moved to in vivo studies. And first of all, we treated wild-type mice with this subchronic administration protocol to check the safety profile of peptide administration. So the peptide was administered at the dose of 3 nanomole gram every day for 2 weeks by intraperitoneal injection. So we check food intake, the weight of the mice, we analyze the serum profile, and all the hematology and biochemistry parameters were within interval reference, so there were not clear toxicity features. We check also target engagement. So we found that in the brain of these mice, there was an increase in ADAM10 postsynaptic localization and an increase in its activity. And then we analyze also cognitive function in these mice. We perform a novel object recognition test. So in this test, mice are in an arena exposed to 2 identical objects. And then the day after, one object is changed with a new one. So if the mouse remembers the 2 objects, we'll spend more time on the novel object. And as you can see, in all the 3 experimental groups, so saline, inactive peptide, or PEP3, the mice were more interested in the familiar objects, so where they were remembering that was new. So it seems that this peptide is not toxic, not affecting cognitive function, so we moved to APP-PS1 mice, so to a model of Alzheimer's disease. We applied the same administration protocol. We check our target engagement, so there is a reduction in ADAM10-AP2 association, a concomitant increase in the postsynaptic levels of the enzyme, and an increase of its activity measured as SAPP-alpha release. But when we perform our cognitive function test, so the novel object recognition, we were disappointed because in these mice that were at full-blown pathology, so 12-month-old mice, there is a clear impairment of cognitive function because the mice were not able to discriminate the two objects, but the PEP-free treatment did not affect cognitive defects, so it was not effective. So we were able to design a tool able to interfere with our target, so ADAM10-AP2 endocytosis, able to increase ADAM10 synaptic activity and, in the mice, but it was not able to improve or ameliorate cognitive function. So we're wondering whether we were treating the wrong population of mice. I mean, probably they were in a too advanced pathology state, so they were not responding to the treatment. So we started to think about treatment mice in the earlier stage of the disease. So we took 6-month-old mice in which we had already the pathological feature, and we check our target engagement. Again, ADAM10 postsynaptic level and its activity were increased. And now we could see that PEP-free administration was able to rescue cognitive defects, as demonstrated by the novel object recognition test and also by another test that was analyzing spatial memory, like the Y-maze. But which is the mechanism underlying this effect of the activation of ADAM10 in the synapses? So first of all, we analyze amyloid plaque burden. We use free antibodies, small plaques, large plaques, whatever, no changes. So it's not affecting the amyloid burden, but what we found was that this treatment was increasing the percentage of mature and active spines. So it was effective on restoring the synaptic and the structure of the synapses, so the synaptic transmission. So more than on a beta load, it was effective in rescuing synaptic activity. So we were able also to test the peptide in human neurons, in neurons derived from iPSC, to demonstrate that it was also effective in human cells. And we found that this peptide is able to increase, again, ADAM10 in the postsynaptic site of the neurons, of the synapse. And this peptide is also able to increase the spine density, both after a short treatment of 30 minutes, but after also 24 hours. So we have a tool able to interfere with ADAM10 association with AP2, blocks the endocytosis, so the enzyme is more available at the plasma membrane, cleaves its substrate, cleaves APP, releases SAPP-alpha, and it has a clear effect on the synaptic structure and function. But the issue is that now, okay, we have to administer this peptide to early stages of the disease. So the next challenge is also for the patient to identify patients that are really early in the disease trajectory, because now it's it's well accepted and recognized that the pathophysiological changes begin many years before the clinical manifestation. So the Alzheimer's disease is a spectrum that ranges from a clinical asymptomatic phase to dementia and to cognitive impairment. And this is also supported by the analysis of biomarkers. You can see that in the preclinical stages of the disease, there is this alteration in amyloid beta accumulation that is followed by synaptic dysfunction, and then tau and neurodegeneration. So supporting also a little bit the amyloid hypothesis. So the first one is a beta, but the second one is synaptic failure. And therefore, now several studies are analyzing synaptic protein in the CSF of Alzheimer's disease patients and in other forms of dementia, because we need a biomarker of synaptic failure to track the disease, to see the changes in the progression. And so we're wondering whether ADAM10 pathways can be useful also to monitor synaptic function and dysfunction in the patients. And since ADAM10 cytoplasmic tail is the biological locus responsible for this trafficking regulation of the enzyme, we carried out a 2-hybrid screening, and we found another protein partner that is CAP2. CAP2 is a member of the cyclase-associated proteins, and these are actin-binding proteins responsible for the regulation of actin dynamics and for vesicle trafficking. But there are 2 members. One is CAP1, that is expressed everywhere, while CAP2 is expressed just in few tissues and organs, including the brain. So we first demonstrated that CAP2 is really a binding partner of ADAM10 by co-immunoprecipitation assay. We detected the complexes of ADAM10 and CAP2 along dendrites. And CAP2 seems to be a regulator of ADAM10 synaptic localization because in CAP2 KO mice, we detected a decrease in ADAM10 synaptic levels. But CAP2 was, let's say, also not really characterized synaptic protein. So Silvia and Lina carried out a full characterization of CAP2 function in spines, in neuronal, in hippocampal neurons. They demonstrated that CAP2 is a postsynaptic protein, but CAP2 is also a protein relevant for shaping the spines, because when you downregulate CAP2, you can see a clear change in spine length and spine width. But CAP2 is also a protein that is reduced in the patients. In particular, CAP2, the total levels are reduced in the hippocampal specimen, but not in the superior frontal gyrus specimens of Alzheimer's disease patients. So this is a specific hippocampal feature of the disease. And the reduction of CAP2 in the total levels of expression leads also to the reduction of the availability of the protein in the synapses. And this kind of alteration have been also demonstrated in APP-PS1 mice. So the mice at the age of 6 months, so early stage, they have a decrease in CAP2 synaptic and total levels in the hippocampus. So we wondered whether restoring CAP2 levels could prevent the synaptic failure that we can see in these mice. So we use a viral vector, so this approach, to overexpress CAP2 under the control of a neuronal specific promoter in the hippocampus of APP-PS1 mice at 4 months of age, so before the pathology onset. And then at 6 months, we analyzed the plaque load and the cognitive function. And we found that CAP2 overexpression prevents amyloid plaque deposition and also the cognitive defects that we can detect in APP-PS1 mice, as demonstrated by the novel object recognition test. So we have a protein that is strictly related to the primary mechanism of Alzheimer's disease pathogenesis, because it is a binding partner of ADAM10, that is the enzyme of the amyloid cascade that prevents a beta peptide formation. But it's also a synaptic protein. It's a protein that can control the spine structure, the synaptic structure. Is a protein that is specifically reduced in the hippocampus, but not in the cortex of Alzheimer's disease patients. And that, as a proof of concept, if we restore CAP2 levels in the mice before the onset of the pathology, we can prevent the amyloid plaque deposition, and we can rescue cognitive function. So we wondered whether CAP2 can be a potential candidate to track synaptic failure in Alzheimer's disease patients. So we measured CAP2 levels in collaboration with Professor Padovani and Andrea Pilotto at University of Brescia. We measured CAP2 level in the CSF of Alzheimer's disease patients and in patients affected by other forms of dementia, such as frontotemporal dementia or Lewy body dementia. And as you can see, Alzheimer's disease patients across severity stages show with higher CAP2 levels compared to control and non-Alzheimer's disease patients. So there is an increase in CAP2 levels in the CSF, also in the prodromal stages of Alzheimer's disease. So in conclusion, I hope to have shown you that the synapse can represent a critical substrate able to link a beta pathology, so the core of Alzheimer's disease pathology with the cognitive symptoms, so that are underlined by the synaptic failure. And that investigating the mechanism in the synapse that are correlated to the initial stages of the disease is important because we can find functional abnormalities that are useful and relevant for developing tools able to tackle these synaptic features, but also maybe identifying biomarkers that are important to follow the progression of the disease or to find the population able to respond to the treatment. And last but not least, I would like to thank the people that perform all this work. So thank to Monica Di Luca and Fabrizio Gardoni, that led the lab of Laboratory of Pharmacology of Neurodegeneration. The green one are the ADAM10 team, so Stefano Musardo and Sebastien Terrain. And the red team are the CAP2 girls, so Silvia Pelucchi, Ramona Stringhi, and before it was Lina van der Meulen that work on the CAP2 story. I thank also our collaborators and all the funding agency and you for your attention.
Thanks, Elena, for your talk. I have two questions. One is about the development of drugs, activators of ADAM10. I mean, is there anything ongoing? I mean, your strategy is beautiful, but is there a path that you see toward drugs that activate ADAM10? And second question, I noticed in your NOR test for the PEP3 that you have a lot of variability in the performance.
Okay.
Is there, and the number of animals that you use is between 7 and 8 per group or more?
Yeah.
But how about the variability?
Okay, so the mouse, I mean, in this slide, this was the original experiment, and the reviewer asked us if there was any difference in male and females. So we have the results in the paper are, let's say, with the double of the mice, and we check also if there was any gender difference, because in this mouse line, anyway, the females develop the pathology a little bit earlier compared to the males. But we found that both genders responded to PEP-free treatment, so they were both effective in ameliorating cognitive function. And about the other question, and FRAX activation, well, we didn't check it, so I don't know. So to me, I know that there are several inhibitors of ADAM10, but apart from previous work using acitrein as let's say, a molecule able to increase ADAM10 expression because it goes on the retinoid receptor, I don't really know other strategies about this.
So thanks, Elena. It was really, really interesting. Curiosity question. So this altered the level of CAP2. What do you think it's the driving force there? So is it something because it gets more produced, it gets less degraded? So what's going on there?
I think it's an issue of expression because we have also a reduction in the mRNA levels. So next question is to understand how CAP2 is the expression is regulated, but this will be, I think, a future issue. Okay, since I'm here, I see no other question. It's lunchtime. I would just like to thank to all of you to be here and to listen to our symposium, and thank to all the speakers for the beautiful presentation, and a nice lunch. Pure on time.